terms of continuity equation. So for compressible fluid, we have the equation, and for incompressible, I will not give much time because my motto is to show my model. So this is the momentum conservation. Again, momentum conservation, you can see this is the left hand side is the inertia term, and the inertia term, again, this is the local local acceleration, and this one is the convective acceleration, pressure terms, shear force, and this is the gravitational or external forces. You may, whatever forces are applicable for the fluid dynamics problem, you may include. So this is the based on the then we have energy conservation. We can also discuss the energy conservation in this. So last speaker have already explained the terms and these things. So you just see, I will come to the my topic, which is very recent topic in the fluid dynamics, known as the electrokinetic phenomena. So what is electrokinetic phenomena? When we study the shearing process, because in fluid dynamics we are studying the shearing process based on the viscosity and surface property, surface, or if any fluid is flowing through the channel, so what is the property of channel? Because channel is your solid and the fluid is flowing through that channel. So what is the property of surfaces as well as fluid? And also there is a shearing between the two fluid layers. So we are dealing like a shearing process in the fluid and solid interaction. But here, if the fluid is an electrolyte solution or aqueous solution, where cations and anions are also involved, and if surface are also negatively charged or positively charged, so there is a shearing process between the surfaces, and force, which force is electric forces which we apply. So we can simply define that if we'll study the shearing process under the effect of electric force, so that phenomena is known as electrokinetic. If we can just a minute, I can show you one video, so the electrokinetic phenomena will be more clear. Just a minute. So I have to stop this presentation and show the my window where I can. So I can. I think is it visible? So you can see this video.
Yes, sir. Yeah. It is visible.
Thank you. Thank you.
I'm fine.
So you can see how if we apply the electric field, this channel is negatively charged, and this electrolytes solution or aqueous solution are having negative and positive charges like cations and anions. So how it is moving, and there is a shearing, and due to this surface, if surface is negatively charged, so most of the positively positive ions are attracted with the surface. So this is the complete process of the this. Let me come to my presentation so that I can. So this is the advantage of because we are dealing the fluid dynamics problem, but most of the, if suppose NaCl is there, NaCl solution is there, but we are simply treating our problem as a fluid dynamics problem, and we are not considering about the electric field are their charges, what are the role of charges. PPT is now visible, sir?
Yes, sir, it is visible.
Okay, thank you very much. So in the under the electrokinetic phenomena, we are dealing four basic phenomena. First is electroosmosis. So most of the people are working on the electroosmosis force when we are studying the fluid motion under the effect of external electric field. And if we are dealing the electroosmosis, so due to electroosmosis there is a generation of potential, and that potential is known as a streaming potential. So as last speaker told you that in fluid dynamics we are dealing two type of approach. One is a Eulerian approach, another is a Lagrangian approach. So if we are dealing the Lagrangian approach, then we have to think about the fluid particles. Okay, so when here also, if fluid particle is charged particle and we are dealing the fluid particle motion of the fluid particles, charged fluid particles under the effect of external electric field, so that phenomena is known as electrophoresis. In the case of electroosmosis, it is due to the Eulerian process, because here we are not considering the fluid particle, we are considering the fluid motion. And due to this, again due to this electrophoresis, if there is a generation of the potential, so that potential is known as a sedimentation potential. So when we are working on the electroosmosis or electrophoresis, so we can also discuss about the streaming potential, what will be the potential and what will be the potential distribution during the fluid flow. So what is the next? What is electroosmotic? As my title is electroosmotic flow of the electroosmotic flow with microfluidics application. So we should also know what is electroosmotic flow. So when a fluid is moving in the channel, if channel is surface of the channel are charged, whether it is negatively charged or positively charged, doesn't matter. And the fluid is a electrolyte solution. Electrolyte solution means it is aqueous solution, means anions and cations both are there. So when we are studying the fluid motion or aqueous fluid motion under the effect of electric field, so that type of flow is known as electroosmotic flow. So what is the difference? Because in hydrodynamics, especially in fluid dynamics problem, we are studying the pressure driven flow. So when we are dealing the pressure driven flow, so the nature of velocity profile is just like a parabolic nature. Means at the surfaces the velocity is low, or it is very close to zero, and in the center of the channel the velocity is the maximum. So due to that nature, and we are also dealing sleep condition and no sleep condition. If it is no sleep condition, then at the walls the velocity is zero. If it is no sleep condition, then there is some sleep velocity is there. So that sleep velocity will be at the surface, and after that the velocity will increase. So at the center of the channel there will be the maximum velocity, and that maximum velocity we consider in fluid dynamics problem, it is maximum velocity. And the nature of velocity profile or the nature of flow characteristics is parabolic. So sometimes it is known as a hydrodynamic flow, sometimes it is known as pressure driven flow, sometimes it is known as parabolic flow. And sometimes when the there is no developing, there is no change in the fluid velocity, then it is also fully developed flow is no, considered. So this is the under the pressure driven flow. Means when there is a channel, and when we apply some forces at the end, like when we pump, when we do some pumping, or when we apply some forces at the inlet portion and generate high pressure at the inlet, and at outlet there is no pressure, like zero pressure, then from high to low pressure fluid is flowing. This is the basic concept about the fluid flow. fluid is flowing from high pressure to low pressure, or simply you can say due to negative pressure gradient it is flowing. In same thing, when we apply the electric field, like this is the electric field, here positive and here negative, and electrolyte is moving. So the difference between electroosmotic flow and the parabolic flow is the nature of velocity profile is a trapezoidal nature, means there is no much variation, and this is due to the electroosmotic flow. And if there is no much variation in the fluid flow, then the analysis is very easier. And also, if you are dealing the biological problem, and if there is no variation in the velocity, that means that is the case of very controlled fluid motion. Okay, very controlled fluid motion. And if the motion or movement like blood flow, if blood flow is very in controlled moving, flowing in very controlled manner, okay, then there is no problem, or there is no case of any diseases. So that is the advantage if you know the electroosmosis, or when you study the electroosmotic flow. So this is the difference between electroosmotic flow and pressure driven flow. So this is the complete structure of this electroosmotic flow, that there is a negatively charged surface, and in between there is a electrolyte is passing, and we apply the external electric field. This is the voltage, and this is anode and cathode. And when fluid is flowing from this portion to that portion, so the nature of that this is electric double layer will discuss in next or next to next slide also. So what are the basic principle when we are dealing any type of physics? Then we have to know the basic principle or basic equation, because without those equation or without those concept, we cannot do any type of modeling, or we cannot go for the theoretical analysis as well as mathematical analysis or numerical analysis of that problem. So these are the basic equation which are applicable, especially in the case of electromagnetic framework, when we are dealing the electroosmotic flow or this. Another thing I can say you, because most of the people are working on fluid dynamics, they are using magnetic, they are showing the effect of magnetic field. So what is the disadvantage of magnetic field when we are doing the analysis of the magnetic field, or magnetohydrodynamics, we can say that magnetohydrodynamics. So with the effect of magnetic, one thing is the magnetic force is always acting in the transverse direction, and the second thing is the magnetic field. Due to magnetic field, we can reduce the fluid flow. This is the standard result of the hydrodynamics. So we can reduce the fluid flow with applying the magnetic field. But the advantage of electric field is we can reduce the fluid flow, as well we can improve the fluid flow. So both side effect, and also we can apply the electric force in direction of the fluid flow, and we can also apply the electric field opposite to fluid flow direction. So both side advantages are there. And when it is required that we have to reduce the fluid velocity, we can do with the help of electric field. And when we have to improve the fluid flow, we can also do it with the help of electric field. So this is the advantage of electrokinetics. And when we are working on this, so using this principle, we can calculate the total force due to this electromagnetic framework. So that is in the form of, this is electric field, and this one is the magnetic field, and this one is an extra term. So we can use these terms, and we can do modeling with the electroosmotic framework. Second phenomena or physics is also related to electrokinetic, which is known as electric double layer phenomena or electric double layer. So it is also important. I just explain with the help of this figure, that if the surface is negatively charged and the liquid are freely moving, so when liquid are electrolytes, when liquid is having, cations as well as anions or positive and negative ions. So if surface is negatively negative, then positive ions will attract on this surface. So all the positive ions will be very close to this surface. So that surface is known as stern layer. So when we see there is a, layer, and this layer is due to negative surface and positive ions. So that surface or that layer is known as a stern layer. And after that, the effect of this negative and the positive ions are lesser, so that there is a distribution or diffusion of the positive ions as well as negative ion through the free surface, through the free surface. So you can see from this surface that there is a distribution of
Disturbance ಆಗಿದೆ. Any problem, sir?
Sorry, there was a network problem due to electric cut.
It's okay, sir. You can continue.
Okay, thank you. So let me again share my PPT. So this is related to electric double layer. So as I discussed that there is a diffusion. So up to some label, there is a diffusion of the ions. So that layer is known as diffusion layer. And when we talk together both the layers, so that is known as the electric double layer. And in this case, we can also see the, potential distribution. This is size the potential distribution with respect to this both the layers. So when we plot the potential distribution across the transverse direction, then there is a, uh, parabolic distribution. And when there is a intersection point with the stern layer and this graph, so that point is known as the potential at that particular point is known as a zeta potential. So when we are working on electroosmotic flow, this zeta potential is also zeta potential plays a important role in the case of electroosmotic flow also. That's why I have shown this, what is the zeta potential. So next is the microfluidics application. I will talk very little about this application. So earlier I just given one comparison with the traditional lab and now microfluidics lab. So what is the advantage of today's development in the fluid dynamics? In the chemical lab, or in the in the case of diagnosis, what we have to do, we have to use two chemicals in large amount, and give the time for the reaction, and then we can find out the output in the form of this. So when we have a large amount of the liquid or chemicals, then it will take the time, and also it will cost effective, because we have to purchase large amount of the chemicals or samples. Okay, but by using the microfluidics devices, what is the advantage? One drop of the chemical or one drop of the liquid, when we use to diagnosis with some chemicals, then one drop is required. So it is also give very fast output, and it will not take much time, and it is also save the time as well as the cost of the chemicals. So this is the advantage of new development in the fluid dynamics. So now people are working on the microfluidics devices as well as the microfluidics chips. So you can see, since my topic is more related to biological flow phenomena, so I have shown some application which is related to biological chips, which is known as human organ on chips. So by using such type of chips, we can control the physiological or biological flow phenomena. So that is the development of a lung on chip. If there is any problem in breathing due to the lungs, so we can also use lung on chip, and we can control the breathing process. So this is the advantage of external chip we can use, and we can control the breathing process as well as other flow which are in biological system. So these are also when we have some microfluidics chips, we can do the sampling process, or we can give the diagnosis of the sample, blood sample or other sample, and we can give report very fastly within the time, so that it will help to doctor to start the treatment of the patient. So these things are also now you can see this is the time of corona pandemic, and there are testing of rapid test. So when you use rapid test, you can have output very fast. Recently, if you heard, one of the my supervisor in IIT Kharagpur, I have worked with Professor Suman Chakravarti. So he has developed, his team has developed one chip which can give the diagnosis of this infected people, and very fast he can able to say that whether the people are infected or not infected with the coronavirus. So these are the advantage of rapid test when you develop, or when we develop microfluidics devices. So we can also have a look basic physiological system where fluid dynamics are involved, and people are working on this biological or physiological flow phenomena by using the concept or principle of the fluid dynamics. So these are the system, physiological system in our body. So everyone knows. I will not give much time because we are not much time. I have more PPT to discuss. So this is the one of the pumping phenomena in the biological or physiological system. When we are eating the food, and from mouth to stomach it is moving, and this pumping process, because due to muscle contraction and relaxation, it is creating the pressure, and due to that pressure, but in control way, it is not due to gravitational force, that if you eat the any food and the food suddenly goes to stomach, it is moving very controlled manner, and due to this trapping phenomena and muscle contraction and relaxation. And based on this pumping phenomena, this is the natural pumping, and based on this natural phenomena, many mechanical people and civil engineers are designing the peristaltic pumps, which are applicable everywhere, even in our home also, to transport or to deliver the water or any type of fluids. So these type of pumpings are frequently used in our real life, and these are very useful. So concept behind this type of pumping is also it is related to natural pumping phenomena. Now another type of pumping is in our body is heart pumping, and this is the complete process how heart is pumping and how our blood is moving from heart to our body. So this is the complete process. That first process is cardiac diastole, then arterial systole, then arterial diastole. So three process, and this is due to the you know uh, internal mechanism involved in the heart, and due to this internal mechanism heart is pumping, and due to this pump our blood is flowing. We are not externally pumping our blood, it is internally pumping, and this is the another type of pump. Third type of pumping in our body is the membrane pumping. So in our body there is a cell membrane, and due to this cell membrane there are a pump, and we can see how it works and how it pushes the fluid, biological fluid, from one part to another part. So these are the membrane motion or motion due to membrane pumping. We have applied this membrane in upper wall only, here in both the walls, lower and as well as upper wall. So this is the complete cycle how to do the modeling. So I will not give much time because we are lacking the time. I don't know how to manage. So I will come to my first problem, that is recently published in Physics of Fluid in 2020. So I will focus on this model. So based on all the basic things, I have developed one model with the help of my student and collaborator. So this is known as electroosmotic flow with membrane pumping. So what we have done, we have considered the surface, lower surface, which is negatively charged, and the liquid in this channel are electrolyte solution, and for upper surface we have fixed the membrane. So how membrane will work, and how membrane will push the fluid, and what will be the effect of electric forces. So everything we have considered in this. So this is the geometry of my model, and we have formulated the equation for propagation of membrane contraction. So we have designed one equation in the form of this, which work in the I have shown this. This is just like contraction. So when we do the animation of this equation, this mathematical equation, we can give or we can find that type of animation. So this is the equation for propagation of membrane contraction, and everything is very simple, and you can see how we can relate our fluid mechanics concept in application of biological flow, and as well as the role of electric field. So what we have considered, this is the momentum equation, as you know. So what external term we have considered, we have used external terms as a electric body force. As I told you that if we have to do the analysis for electric field, then we have to use some external force like electric field. Then for electrokinetic phenomena, we have to use Poisson equation, Boltzmann equation, because ion distributions are there. Then we can consider both the equation together in one equation, that is Poisson Boltzmann equation. All these things are very basic, nothing is new, but with the help of these basic things we can discuss, or we can analyze the new result, which is very applicable in the biomicrofluidics application. So process is same. We have done non-dimensional analysis with the help of scaling process, and we have converted our governing equation. These are the governing equation, and we have converted our governing equation in non-dimensional form. And then we have to, in fluid dynamics problem, we have to also define the constraint, or we have to define the boundary condition, so that we have taken the boundary condition u is equal to 0 at the lower wall. H1 is a lower wall and H2 is a upper wall. So this type of boundary condition we have used. We have also find our solution in the terms of some special function that is known as Polygotham function, because when we are dealing the partial differential equation, many people know that solution we are generally find in the form of Bessel function, modified Bessel function. So some other special functions are also there. When we solve the system of partial differential equation, we can have some special function also. So when we solve the governing equation subject to boundary condition, we have basic fluid properties like axial velocity, transverse velocity, potential function, stream function we have calculated and obtained the result in the form of wall shear stress, pressure gradient, volumetric flow. Everyone knows how to solve the partial differential equation, so no need to discuss every step. We can have solution, and other constants are also involved. We can note down. So these are the constant. Now we come to, so we have used Mathematica software as well as MATLAB software. Sometimes some results are very good in coding of when we are doing simulation in MATLAB. Sometimes it is very good in Mathematica. So we have to know both the software, and it is also advantage if you know two softwares, coding in two softwares, then you may also verify your results, so that if results are same in both the coding, you have written code in Mathematica as well as MATLAB, so you can compare both the results. If results are very good and also both results are same, then you can say that yes, our coding is right and the results are also right. But the validation of the results is different part. You have to validate your results with some existing result, or we have to validate our results with some experimental data. So if experimental data is available, then we can discuss validation part with the comparison of our numerical results with the experimental result. If it is not available, you may check some existing result. Numerical results are available in literature which is published in very good journal, then you may compare with that. And if your solution are analytical solution, closed form solution, so there is no need to compare, because closed solution are always exact form and no need to compare your result with others. So that is always correct. Okay, so this is the uh, distribution of the potential. This phi is the potential, and we have seen the potential distribution with different zeta potential. So you can see how it is distributed along the transverse direction. Second is also this kappa is the inverse of ideal thickness, electric double layer thickness. So it is also important. We have to, whatever key parameters involved in your physical problem, all effects you have to analyze and discuss. And after discussion should be very you know uh, close to related to the physical interpretation, because discussion is not like this zeta potential function is increasing with increasing the zeta potential, decreasing with the zeta potential. No, you have to, if it is increasing, why it is increasing, what physics involved in this increasing, why it is not decreasing. So that type of discussion you have to do. And when you have you will do such type of discussion in your paper, definitely your paper will get published in very good journal. So this is the way. So presentation, discussion, and the way of writing the paper are very important. If your results are very good but your presentation is not good, then you will not get good publication. So you have to learn and the quality of figures, quality of results you can see. So can I take 5 to 10 minutes, sir?
May, because 4:40 means buses are there.
Okay. So I will go very quickly
Try to go quickly, sir.
Sure. So I will just show what are the results. You can see this is the pressure distribution with this is the electric field effect. So we have taken negative electric field as well as positive electric field, and the UH is equal to 0 means there is no electric field. So we have taken all the cases that if electric field is applied in the direction of fluid flow, if it is applied opposite to direction of fluid flow, if there is no electric field, then what will be the pressure distribution. We have also seen the streamline, what will be the streamlines for this type of fluid flow. So I will go very quickly. Then shear stress distribution, then velocity profile, because velocity profile is also important. So with respect to all the parameter we have discussed, so these are other velocity profile. Then flow rate is also sometimes people are saying flux. So these are the flow rate, inlet flow rate, outlet average flow rate. So all type of discussion. So this is the conclusion. So I can conclude basic things. So any type of physiological flow or industrial fluid flow can be controlled with the help of electric field. So I have prepared others models also, but due to time I cannot go. So you must just see the other models. Just 2 minutes, sir. Particles trajectory also we have discussed with the help of Lagrangian approach, and other embryo
Electroosmotic
transport we have discussed many things. So I will not, due to lack of time, you can see the results, how we have simulated the results, the quality of results, so that it is very useful. And one, international school I am going to, organize in month of January. So those who are interested may get registration. So you just see the speakers. It is international winter school on fluid dynamics and heat transfer. So most of the renowned professor related to fluid dynamics and heat transfer have been consented for this school. So those who are interested may get registration. Fees is also very nominal, 500. If it is TICUP sponsored, then there is no fees. So thank you very much for listening. If there is any query, you may discuss. Otherwise, later you may ask through my email ID. Okay, email ID is there with Professor Markande.
No, sir. It was a very wonderful session, sir. I'm happy that today's of these 3 days, today's afternoon sessions were real FDPs. Otherwise, other sessions were just like they were conference sessions. They have discussed about their paper and they have discussed about their work. But you have discussed in general, in depth flow dynamics, and even the first session was also very good. So I'm really happy that today's sessions were useful even for the faculty who are interested in teaching fluid dynamics. Really, it's a wonderful section. Now, over to participants. If you want to clear any of your doubt, you can. I'll give you 2 minutes time. Those who want to ask questions can ask the questions.
Sir, good evening.
Good evening,
This is Dr. Mohan Krishna, a
sir.
professor from Vidyavardhaka College of Engineering, Mysuru, Karnataka.
Sir, please.
Respected Dr. Dharmendra Tripathi, you have delivered a wonderful talk.
Sir, I am not respected. I am just colleague. Your colleague. You tell Dharmendra.
As a salutation, we have to use the word respected. So it's okay, dear Dr. Dharmendra Tripathi. You have delivered a wonderful and resourceful talk. Anyhow, the talk on electroosmotic flow, definitely it is a need of the hour. Sir, other than IIT Kharagpur, where exactly the research scholars can find facility for experimentation pertaining to this?
Sir, actually, most of the student of Professor Suman Chakravarti, they are working in IIT Delhi in mechanical department. They are all working in IIT Guwahati and
Okay, sir.
IIT Ropar. They are also working because they are also my colleague. So they are developing the experimental facility there only. So you may get.
So once when you refer IIT Ropar, definitely it should be Dr. Sarit Kumar Das, the
Yeah,
IIT director.
yes. Sarit Kumar Das is also there.
Okay, okay.
So he's working in electroosmotic and nanofluids.
Okay, sir.
So
Okay.
he has developed very good lab there. So if you are
Okay.
interested, because my interest is only simulation and mathematical modeling.
Okay, sir.
So
As said by Dr. Markande, anyhow, this session was absolutely mesmerizing. You have given wonderful information pertaining to this. So we should be really grateful in delivering a very meaningful talk. Thank you very much, Dr. Dharmendra.
thank you very much, sir, for your appreciation.
Thank you, sir.
Thank you, sir.
Thank you, sir. So with this, I hope no more questions or no more inquiries, no more queries. I mean, so
Even
if you have got any queries, you can post it in the WhatsApp group. I will pass it on to Dr. Dharmendra Tripathi. Not a issue.
yes, sir. I will be happy to discuss, sir. No problem.
Yeah,
Thank you very much, sir, once again for inviting me and
yeah.
giving opportunity to interact you people.
Yeah, really, it is a wonderful session, sir. I am also a biomechanics man, so certainly I was very much fascinated by your talk. So the talk should be like this. That's what I felt. FDP session should be like this. That's what I felt. So I hope even my participants also are on the are of the same view. So with this beautiful session, we are going to end today's day 3 session. Thank you, sir. Thank you.
Thank you very much, sir.
Thank you, sir.